What it do, class, and welcome back to another day of Mr. Fitz's biology class. Today we're diving into passive and active transports. So before, we've already talked about types of solutions, we've talked about the cell membrane, we've talked about things moving. Now we're going to talk about how we can actually transport those things that are moving. Just a real quick review for you. We did talk about the different types of solutions. So you have hypotonic solutions on the left there, isotonic in the middle, and hypertonic on the right. Notice in that hypotonic, think of those dots as solutes, or something that's dissolved in water, like sugar or salt, okay? And more specifically, we're going to think of them as a concentration or a percentage or amount, okay? So concentration is just the percent of stuff that's dissolved in water. So that far right picture over there, the far left picture, hypotonic solution, is a kind of like 1% dissolved, because there's only one dot there. So think of it as a percentage. And then on the inside, it's got like 6%. So water will flow inside this one because I have 99% water and I have 94% water in here. And the reason I know the water percentages is I have 1% that's dissolved stuff. That means the other 99% is water. If I have 6% that's dissolved stuff, that means the other 94% is water. So that's the reason that water wants to flow in versus flow out. In an isotonic solution, you have equal concentrations on each side, or equal percentages on each side. Therefore, the water amount will be equal on each side as well. That's why water will flow both in and out of the cell simultaneously in a dynamic equilibrium. And then finally, with a hypertonic solution, you're going to have more concentration, more percentage of solute outside the cell, a lot greater number, there's about 20 here, than inside the cell, which is 6. So that means that's 94% water in here, the other remaining percentage is water, so 94% would be water inside the cell. And in this case, because 20% is solute, that means the other 80% is going to be water. So in this case, water will flow out of the cell, going from where there's 94% to where there's 80% to try to spread it all out and get to an equal number over time, okay? So you do need that kind of awareness as we talk about these transports, because it does make a little bit more sense. And I didn't know if it was clear from the last video, so I wanted to highlight it again before we got a little deeper. So the big questions that we need to answer are, what are the types of passive transport? There's actually multiple types of passive transport, and there's only one type of active transport, so it's just active transport. And then what is the difference between those two things, active versus passive? That's actually a pretty simple question. It seems complicated, but that one's pretty simple. And then this one's going to require that you are very organized with your note-taking to know which ones are the passive transports and then which one is the active transport. Okay, so let's start off with passive transport. These are actually ones that we went over in the previous video where we talked about the movement of particles, osmosis and diffusion, where the particles just naturally want to move from areas where there's high concentrations to areas where there's low concentration. So they naturally want to move that way. They just want to spread themselves out. So the cell does not have to put any energy in. There's no requirement of energy. That's what actually makes it passive. It's just kind of sitting and letting this naturally occur. Okay? So they're going to have these particles that move through the bilayer, or molecules that move through the bilayer, and they're very specific ones that can. So water, for instance, can easily move through. That would be the osmosis part, right? Also, gases like oxygen can easily move through, and carbon dioxide, because we want to make sure that we're getting oxygen inside the cell. It's small as a molecule, so it can squeeze in these gaps, and carbon dioxide can squeeze on the way out to allow a nice exchange of gases to kind of flow. And usually what happens is you're burning oxygen inside your cell, so the oxygen wants to flow only in. You usually don't have a surplus in your cell. It's constantly burning it, so it's going to pull more in, and it's going to constantly be producing CO2 down here, so it's going to release it back out. So it's just all based on, there's a lot of oxygen out here, there's less oxygen in here, so the oxygen is going to move in. There's a lot of carbon dioxide in here, less out here, so carbon dioxide is going to leak out. It kind of spread out just naturally, kind of fill out the room. Okay. Now, another thing too is that we do have these proteins that run through the membrane. Okay, so these proteins that go through the membrane, they do have specific jobs. So really, why are they there is to transport materials. Okay, these proteins are known as transport proteins because they give either doorways or they give channels for particles to move through that are maybe a little bit bigger than oxygen or carbon dioxide or water, but the cell still needs to either move them in or out. So The way that this one works is called facilitated diffusion. Facilitate means to help, and diffusion means to just naturally spread out, right? So we're just helping the natural spread. So in this case, we have these channel proteins. This is a great example right here. It's got a nice little tube through it. And that will allow particles to flow both in and out across it. Now, they're usually designed to kind of only flow one way, so that way we don't have a backlog, but it does allow movement of particles. So for instance, I have more solutes here, more circles outside the cell than I do inside. So that, therefore, diffusion says that they want to go in the cell, but they're too big to fit through the membrane, so they have to go through this transport protein, this little channel, to be able to actually fit through. Okay, so these proteins are kind of like opening doors to the cell. Usually it's kind of a one-way movement through there because again, typically we have more of this stuff outside than we do inside the cell. A great example of this would be like glucose or sugar. Usually when you eat, you produce a lot of sugar in your blood that's outside your cells, and then they pull that in because you usually don't keep sugar very long. You either store it in a molecule called glycogen, or you'll break it down immediately to use it as energy. And that's how you produce that carbon dioxide. So you're pulling this sugar in constantly into your cell. So anything that can't really just pass through the membrane by itself, that gets kind of pushed away by the cell membrane, we'll have to use these doors or another way to get into the cell. So the reason that these big particles can't go through, again, they won't fit, but also sometimes these particles will have charges to them. And so things that are heavily charged like sodium ions or chlorine ions, those very big positives and negatives, much stronger than water, will not be able to get through because this region right here, this hydrophobic region that is not charged, non-polar, will actually push those things away. So it actually forces them to use a protein that's specifically designed for them to go through. All right. So remember, this actually gives that semi-permeability to the membrane because it's selecting some things that are able to come in and out. It does sometimes provide doors for certain things to come in and out, but not everything is freely able to move through the cell membrane. That's why it's selectively or semi-permeable. All right, now active transport. Now it's important why I actually put the A, T, and P in purple, and I'll explain that in a moment. But active transport is something that you need proteins for because it's gonna require some bigger thing to be moved or a charged particle to be moved. But it needs an energy source, and that energy source is by the name of ATP. You're gonna learn a little bit later that's called adenosine triphosphate, but right now we'll just leave it as ATP. And please know that is the energy money of the cell. You can't do anything unless you have ATP. So that's why I've highlighted the A, T, and P. You can see active transport tells you it needs energy, ATP, all right? So this moves particles actually against where they normally want to go. So normally these particles would want to just flow out because I have a lot more particles inside the cell than I do out. So diffusion would say they should spread. However, sometimes you have some buildup of stuff in the cell that you don't want, and there's less of it outside, so you have to physically move it. So that is where active transport comes in. Instead of it necessarily going out of the cell like it normally would, where it would try to spread out, we're in this case gonna pull it in. Okay, I want to take this thing that's outside the cell that I want inside my cell, and I'm going to pull it in, even though I have a lot more out there. You can flip-flop it if you had a lot of particles outside the cell and only a few inside the cell. Sometimes you want to get rid of those particles. They could be waste or something like that. So you use active transport to move that. So active transport requires energy. Osmosis, diffusion, and facilitated diffusion are your passive transports, and they don't require energy. So really what makes active and passive transports different is if they need energy or not. Active, energy; passive, no energy. All right, and just a reminder in case you've missed it, is the transport proteins do help move things across the cell. So in this case, we're literally pumping things across the cell, pushing them against where they want to go. In facilitated diffusion, we're just opening a door to allow them to kind of frolic through, if you will. All right, the final bit here is a great example that always comes up on test quizzes, the EOC, everywhere I've seen it. This is the sodium-potassium pump. So usually when you're getting asked a question about active transport, this is the example they use. And the reason why is because the cell is trying to move either sodium or potassium where it doesn't naturally want to go. So for instance, the cell always wants to take in potassium. These Ks right here, it wants to bring in. There's not as many out here, so it actually ends up pulling them in. It also has excess sodium. Your cells build up sodium pretty quickly, so they want to actually get rid of it very quickly as well. So they use an exchange here where they push a couple out and they pull a couple in, and that requires energy. You see that ATP down there? That is your energy for this one. So the cell is actually moving these ions, this sodium ion here and this potassium ion here, against where it wants to go. There's already a buildup of potassium inside the cell, but your cells like it, so it tries to pull more in. There's a buildup of sodium outside the cell, but your cell doesn't want to keep it, so it keeps pushing more out. So again, it's moving it against where these things want to naturally go. If sodium was given the choice, it would flow back. If potassium was given the choice, it would flow out. But we are physically, in our cells, pumping these things where they don't want to go, using energy to make that work, that ATP. That's all I have for today. Hopefully this video was sensible to you. If not, please pause, rewind, do what you need to get your notes down, because this stuff does come up pretty frequently on tests. Other than that, have a great day and keep it real.